Thank you so much, Dr. Capps. Appreciate that. It's great to hear from you. And it looks like Sally has the recording going. So we're actually going to figure out how to live Zoom broadcast this to our YouTube channel. So this is the first time we're trying this out. So hopefully everything goes well. Now to the main event. As has been our tradition, our annual McCrone Awards reception is a time to showcase the scale and scope of research and creative activities at Humboldt State. We're excited to bring you tonight 4 TED Talk style presentations from our 2021 award recipients. A little bit of housekeeping for tonight. So throughout the presentations, if you have a question for one or more of our presenters, please use the Q&A function at the bottom of your screen, or you can send a chat through the window as well. And we'll try to select 1 or 2 questions to ask our presenters at the end of each one of their slideshows. So let's get started. First up, I'd like to introduce Dr. Carrie Byrne from our Environmental Science and Management Department, who's going to join me on our virtual stage to help introduce our first speaker, our graduate student. Hi, Carrie. Good to see you.
Thanks so much, Casey. This is the first time I've been on a webinar, so hopefully everyone can see me now.
So far, so good. Take it away.
Okay, great. It's my distinct pleasure to introduce Sean Stewart, who's the recipient of the 2021 Alistair and Judith McCrone Graduate Student Fellowship Award. So Sean is a first-year graduate student in the Natural Resources Graduate Program here at HSU. And he returned to school. He actually had a really successful career as an architectural designer, project manager, and rafting instructor at College of the Redwoods before he returned to school a few years ago to earn his undergraduate degree in environmental science and management here at HSU. And I first met him then a few years ago when I was luckily assigned as his academic advisor. And when I first met Sean, I was immediately struck by his motivation, drive, and passion for conservation science. And I was lucky enough to recruit him as a graduate student into my lab after he graduated from undergrad. He's undertaking a really rigorous and important research project for his master's degree, which he'll tell us about today. So please join me in congratulating Sean Stewart.
Thank you very much, Carrie. The words were very generous. Let me share my screen. Everybody see that? I'm going to say yes. Okay. So for my thesis, I am going to be building population models to quantitatively quantify extinction risks and possible influences on population growth of the endangered plant Applegate's milkvetch. In this presentation, I'll be discussing some brief stuff about biodiversity and conservation before I jump right into my thesis. So first off, biodiversity. So it's really just all the plants and animals, yes, even the insects and microbes that live in a specific area. Now, we're fortunate that Humboldt actually we're in a biodiversity hotspot. So that's pretty awesome. But why is it really important? So first off, we're just all part of one larger food web. If a species goes extinct, that web gets a little weaker. You can kind of think of the loss of milkweed plants on monarch butterfly populations. Second, a lot of species provide ecosystem functions. So in this picture alone, you could see plants converting carbon dioxide to oxygen. You got some mushrooms over here breaking down organic matter, and you got trees that are providing shade for cooler water temperatures. On a more human-centric level, we've got some aesthetically pleasing species like the redwoods, and culturally important like the Humboldt Godwit days. And furthermore, species are used as natural resources, such as raw building materials and food like this delicious chanterelle mushroom. Now, there are a lot of threats to biodiversity, but long story short, humans are the biggest threat. Luckily, there are a lot of people that are trying to conserve biodiversity, and many of them have found a home just like me at HSU. So what is conservation? It's really a term that covers a variety of tools and practices for protecting and restoring species, mostly endangered and threatened species. And of course, it should be based on the best available scientific knowledge. Now, some examples of conservation, at least hands-on examples. We've got invasive species removal, like the yellow bush lupin here and the lupin bash, protecting the wildlife, and planting native species, no matter what scale the project is at. Now, there are a variety of conservation techniques, but we really have to keep in mind that they are species and habitat specific. So for species that we really don't know a whole lot about, we use what is called adaptive management. And that's really changing the conservation practices depending on the changing environmental conditions and updated scientific knowledge. So scientific knowledge is gained typically through the long-term monitoring of individuals, collecting demographic data, and analyzing population viability. So now let's get into the target species, Applegate's milkvetch. So this plant is endemic to the Upper Klamath Basin up here in Oregon. And more specifically, it's only found in and around Klamath Falls. It is a nitrogen fixing legume, meaning it takes atmospheric nitrogen, converts it down into the soil so that other plants may use it. It is a perennial that loses all of its above ground biomass over the winter, and its stems crawl along the ground from a single base. It has these beautiful, small white to purple flowers, and they are food to pollinators and native herbivores as well. The US government and the state of Oregon have both listed Applegate's milkvetch as endangered. There are currently 7 known populations of varying sizes, and it is considered one of the rarest plants in Oregon. In 1998, the US Fish and Wildlife Service wrote this recovery plan, and in it, its ultimate goal is to really downlist the species from endangered to threatened. Since then, there's been 2 transplant attempts. One was not very successful, and that's probably due to its growing environment. And the second one is ongoing. And so that's really where my thesis begins. In 2014, Dr. Carrie Byrne and her students were tasked for monitoring and collecting data from 4 native populations and the 1 transplant population. Through the years, we've marked each individual and collected the life stage and size, as well as the morphological traits such as the number of stems, inflorescences, and fruits of each individual. Furthermore, we've also collected species composition, species neighborhood composition, and that's really so that we can see if any other plant species influence the survival of each transplant individual. So what am I doing with all that data? Well, I'm creating these population models. With these population models, I'm trying to, I will forecast extinction risks and hopefully correlate some environmental conditions such as drought to population growth or decline. And also I will be analyzing the transplant project to see whether or not it's successful or not. The purpose of my thesis is to assist the US Fish and Wildlife Service and other environmental agencies in protecting this plant, and to be able to apply adaptive management techniques. I also am, adding to the long legacy of protecting and conserving biodiversity for future generations so that they can enjoy it, and hopefully they can take up the mantle. So now I'd like to just show some preliminary findings. These are very rough so far, but pretty exciting. So the first one is a population growth map. So everything above Lambda 1, the population grew. Everything below Lambda 1, the population declined. So as you can see, this population spiked in 2019, but overall has declined. This is one of the life stage models that I've created. And in this, you see vital rates. So you can read this as 47% of seedlings become reproductive the following year, 17% become vegetative, 1% go dormant, and the rest die off. And down here, we're showing the reproductive rate. And so what this means is each reproductive individual created 0.36 seedlings on average the following year. This model here was really done through using a 4x4 matrix here, but this is of course a little too simple for me, so I decided to do something a little tougher, and I started creating these 50x50 matrices based on size and growth. So how you read this is the diagonal line here is an individual in stasis. Above it, the individual has grown. Below it, the individual has shrunk. And as you can see, the largest individuals typically shrink. And I just think that might be because they're spending so much energy on reproducing. Now, here's another kind of fun graph. I have projected populations 20 years into the future, and I've done that 100 different times. The dotted line here is the current population size. And so the majority of the time, the population is in decline. So that's really all I have to share with you right now, but I would like to thank all these people listed here, but especially the Dr. Alistair and Judith McCrone Fellowship. You guys really show that you care, and I really appreciate the financial assistance. Thank you.
Thank you so much, Sean. That was fantastic. We did have one very brief question from the audience that just said, what is Lambda? So I'm not sure if that's enough of the question or if you need them to elaborate.
So Lambda is really a way for us to, so Lambda 1 is really a population in stasis, meaning it's not growing or anything. And so instead of using all of the individuals and seeing how it grows that, we calculate it and it's used as just a general lambda.
All right, thank you.
I hope that answered it.
And I heard you mention lupin bashing. Do you know if that's still an opportunity for like community volunteers? I know that used to be a really big push around here to get community members involved with the conservation efforts.
So I've done it 2 years. I don't know whether or not they're continuing it right now, but it's certainly a very good group effort there.
Fantastic. Well, thank you again, Sean. It was wonderful to have you, and congratulations on your award.
Thank you.
Okay, so next up, our speaker. Since arriving at HSU in 2018, biology professor Dr. Catalina Cuellar-Gempeler has earned widespread recognition for her expertise in the fast-growing field of microbial ecology. She heads a cutting-edge research program at HSU that combines modern lab techniques, advanced programming languages, and traditional ecological fieldwork. In her short time here, Dr. Cuellar-Gempeler has mentored 19 undergraduate students, served as the major advisor for 4 graduate students, and published 6 journal articles. And most recently, she secured a prestigious National Science Foundation, almost $1 million grant to research the effects of colonizing microbes on carnivorous plants. Please join me in congratulating Dr. Catalina Cuellar-Gempeler. Welcome, Catalina.
Casey. Hi, Casey. Thank
Hi.
you so much for that wonderful introduction. I'm very excited to be here.
Take it away.
Awesome. I can do that. Let's see if I can share my screen with you, and maybe I can get a confirmation that you can see it. Yeah. All right. That looks like it does. Okay. So I'm very excited to be here. I'm very honored by this recognition. It's really, I'm speechless almost of being here surrounded by such wonderful and talented researchers. And I really just want to take this opportunity to tell you a little bit about what we've been doing in my lab regarding microbial biodiversity and function as foundation for human and ecosystem health. But before we start and before I begin, I wanted to remind you, every single one of you, my friends, that you are covered with bacteria and that we all live in a microbial world where since birth we are completely surrounded, colonized, and in interactions with microbial organisms. And numerically, they outnumber us. There's between 3 to 10 times more microbial cells on you than human cells at this particular time. And functionally, they can also play an important role, representing up to 1/3 of the metabolites in your blood. So this indicates that at some point we're even more microbial than human, and that to understand them is to understand our own life. And it's so very exciting and new because in the advent of new technologies for sequencing and computation, it's that we have been able to look at this diversity and their function. Particularly, there's evidence in the past decade that support the idea that host-associated microbial communities, not just with humans, but with all animals and all plants, provide support to our survival and reproduction. And this is through 4 particular functions that they perform, which include access to nutrients, defense against pathogens that try to attack us, developmental cues that allow us to reach maturity and reproductive stages, and sometimes extended phenotypes with traits that we could not do otherwise. So hopefully I've got you thinking about the types of microbes that live on us, their function, and their relationship with our well-being and our health. But I'm going to push this a little bit further and tell you that, in fact, microbial organisms also play an essential role for the health of the Earth by playing important pathways in the biochemical cycles on Earth, including specific transformations within the nitrogen cycle that no other organisms can do on Earth, driving large proportions of the carbon cycle, and playing important roles in sulfur, silica, and other cycles. So hopefully you're joining me now in thinking that we need to understand who are these microbes, how many are they, and how can we understand the contribution of that diversity into function. And to understand this, in my lab, we go and draw information from theory in ecology. And this idea of linking diversity to function has been around for several decades. And central to this idea is a positive relationship between diversity and function, which posits that as you increase diversity, you increase function. In the shapes of these graphs would look something like this. This theory also tries to understand the mechanisms behind it. So for example, as you increase diversity, you're going to add species that perform different functions and thus occupy the ecological space better, utilizing resources better, and increasing the ecosystem function. In addition, when you increase the number of species, you also increase the odds of adding species that are very functional, and thus leading to the selection of communities that are very functional and diverse. A slight departure from this positive relationship is the idea of redundancy that posits that if you increase diversity at one point, you're going to start adding species that are functionally redundant or function similarly, and so your function is going to plateau. This is nice because it results in communities that even if they lose species, they're going to maintain a stable function in the ecosystem. What we found is that when we try to apply this theory to microbes, these are not the only shapes of this relationship that we find. There's a variety of non-traditional and non-positive diversity function relationships that come to light. And one of the examples is the one you've seen in the screen right now, the negative selection. When you increase diversity, resulting in an initial increase in function that peaks early and then drops dramatically. And this happens when the function is performed by specific species that are typically poor competitors and thus are not able to survive or perform their function as you increase the number of species. So what we propose in my lab is that we need to step away from the idea that we need to find one general shape for this biodiversity ecosystem function. And instead we need to ask, when are we going to find each of these shapes? And by finding this, we will be able to better predict changes in function and diversity and manage changes of function and diversity in microbes or on plants or on animals or in any function and diversity metric that you want to think about. So to tell you a little bit more and kind of like land this idea in an actual biological system, I want to tell you about one of the hosts that we work on often in my lab to test these ideas. So I'll go over a case study of the pitcher plant associated microbial communities. And many of you may be familiar with these plants. These plants are carnivorous plants that produce modified leaves that capture insects that are attracted by nectar and then fall in these traps, these leaves shaped like pitchers or jars, and drown in this fluid and become the food for these plants. These plants use this nitrogen as supplementary nutrition since they live in soils that are typically depleted in nitrogen and other nutrients. Importantly, these two species, Sarracenia purpurea is the purple pitcher plant common to the east coast of the United States, and Darlingtonia californica, which is our endemic pitcher plant in California, are both limited in the amount of enzymes that they can produce. In fact, they produce little to no endogenous enzymes, and thus they resort to the microbial community to actually be able to degrade this insect prey and provide this fitness effect to access to nutrients. So this provides a really nice system for us to look at the microbial communities within the leaves, their function as they're able to degrade the prey, and the resulting host effect and access to nutrients. And we look at this with the idea that we can partition some of these functions by looking at the broad idea of how is the bug broken down, but also specific pathways of how carbon is moving through the system, important to the coexistence of microbes, and how nitrogen is moving through the system, which is very important to understand the relationship between the plant and the microbes. So let me show you a little bit of what we've been finding, and this is just a little snapshot of what we are doing. So what I'm going to do here is show you a variety of results that look a little bit like this graph that we talked about earlier, where we have diversity in the x-axis, like here, and then an ecosystem function on the y-axis. And what I'm going to ask you to do is look at the data that I show you and try to fit it with one of these shapes. And let's see if we agree. Maybe you guys are going to discover something special today. And in this case, we're going to measure function by taking the weight of an insect, and then putting it into the pitcher plant, letting it degrade for 7 days, and then measuring the weight again. So the function that we're looking at is weight change, right? So high function would be up top, low function would be at the bottom, and we have species diversity in the x-axis. Are you guys ready to interpret this figure? Yeah. I'm going to quiz you at the end. All right. So what we found was that the relationship between weight change and degradation ability drops with species diversity, which is fitting with the negative selection function common to other functions in microbiology. This means that this function may be provided by a few species that are able to provide key transformations in the degradation of the bug. Right now, we're working really hard at identifying which are these species, which are these functions, and how can we identify this as it varies across the range of the plant. But this is not where we stop. We think that understanding this is just the first step, because ecological processes around all the range of these plants may be influencing and changing the shape of this curve of this relationship between diversity and function. So what we want to do now is think about how different ecological processes change this relationship. And what am I talking about ecological processes? I'm talking about environmental conditions, fluctuations, nutrient levels, succession over ecological time, interactions between predators and prey, dispersal, and others. You can pick your favorite ecological process, plug it in here, and I think you can find something interesting. And the key here is that some of these processes are going to couple these two relationships, are going to make function and diversity increase together or decrease together. And some of these functions are going to decouple them, sending function up and diversity down, or the opposite. So separating them in the space. So I want to tell you, and this, you could ask this for any function in any system, in plants, in animals, in fungi, in anything. And what I want to tell you today is a little story about how succession influences function and diversity in, you guessed it, pitcher plants. So in this case, we're going to look at Sarracenia purpurea, which has a nice development of a successional process over time. Initially, new leaves open and are sterile. They begin to accumulate fluid from rainfall, typically, or flooding, and microbial organisms within this fluid. As the leaf ages, the tissue becomes a little harder and darker, and the microbial community changes dramatically. So what we can ask is how the microbial community changes in diversity and how its ability of degrade the prey changes over time. All right, let's look at what the results are. So in here we have a figure that looks a little bit like this. We have time in the y-axis and function or diversity, and we want to ask whether these are coupling the relationship or decoupling. Again, we have weight change in the y-axis. And what we find is that the function increases over time, with old leaves being a lot more functional, which, you know, is the first part of the story. The function of the community increases over time. So now let's jump to look at diversity. And in this case, we have age in the x-axis and the species diversity in the y-axis, here represented by the log of the number of ASVs, which is a representative of species from identified DNA sequences. And what we find is that diversity decreases with the age of the leaf, which suggests that function and diversity are decoupled by time. This means that if we want to manage a system that looks like this, if we want to understand and predict how it changes through time, we need to take into account the fact that over time, these processes are changing and the relationship is changing. So I want to jump and leave you with some questions, because that means that you're going to pay attention to what we keep doing in the lab in the next few years. But I want to introduce you to some of the other systems that we look in the lab and ask the same questions. And some of this work coincides with collaborations with faculty at HSU and across the country. And I want to ask Sean for such a wonderful introduction for the first part, which is the Astragalus applegate conservation portion. We are working with Carrie Byrne and their lab into understanding how microbial communities associated with the roots of this plant can contribute to the success of these transplants and the restoration and conservation of the plant. So can we use this information to understand how microbes provide functions to save species like Astragalus applegate? Another example is a collaboration with the University of Alabama, where we're looking at how microbes can provide traits for coastal species to become more invasive. And this is in collaboration also with two of my graduate students, Jonas Sotingo and Parker Lund. And lastly, a system that is departing from our host-associated theme, and it's looking at the constructed marshes in Arcata and asking how microbial diversity and function contribute to optimization of water quality in constructed marshes. And this is in collaboration with the Arcata Marsh Research Institute and Sandrine Thompson, one of my graduate students. And the last thing I want to mention here is that it would not be possible without graduate and undergraduate student research who bring ideas to my lab, new systems and new ways to apply some of these function diversity ideas. And this is where creative science really happens. So I want to close this talk and hopefully you're walking away thinking a little bit about how biological systems, and not only microorganisms, but all biological systems can be thought of as a relationship between diversity and function. And the better we understand this, the better we can predict and manage what happens around us. That these are dynamic systems. And if we don't think of them as dynamic, we're not going to be able to productively interact with them. And lastly, that we can only reach generalizations if we bring many brains to the table with different voices, different backgrounds, different faces. And by coming together, we're going to be able to understand the world. And with that, I want to acknowledge HSU for giving me a home to play with bugs and microbes and have fun. All of my collaborators within HSU, across CSU, and across other institutions in the country and abroad, my students, the graduate and undergraduate students that do so much work and bring so many interesting ideas, and funding sources that include my postdoctoral funding that gave me time to think about some of these ideas, internal funding at HSU that allowed me to jumpstart some of this research, and the NSF Career Award that is going to push this into the future. And to you for listening to me, and your microbes for being there for you. And with that, I'm super happy to take any questions that you may have.
Thank you so much, Catalina. That was fantastic. We do have lots of questions from the audience, so I'll pick a couple. And I'm going to read them, but I'll send them to you in the chat as well. So the first one that we had was, do we know how similar in terms of the composition the microbial communities are in those two types of carnivorous plants?
Very good question. So we haven't, I want to preface this by saying I haven't run the sequencing in parallel in a single project. So I haven't compared them directly. But if I look at sequences from separate projects, I can tell that there's some similarities and some differences, with the particular differences that Purpurea, so the eastern one, seems to have more species than the Darlingtonia, but their function seems to be similar. So some interesting thing to look at. My project starting this summer and continuing into the next summer is going to look at this directly. So maybe let's chat in 2-3 years and I'll have a definite answer for you.
Nice. Another question was, what have you learned about the microbial communities in the Arcata Marsh?
Ooh, very good question. So also you're going to have to wait for that one. The functional aspects are really cool. They are mostly, and if Sandrine is in the audience, she could probably say this a lot better than me. This is all of her work and her ideas. So the patterns are consistent in function with what the Arcata Marsh Institute has been finding throughout the years. They've been taking very reliable data on the functional aspect and how it changes across the ponds. So basically what you have to take into account is that the treatment facility dumps the water into the oxidation ponds, which are the ones in the far end, and then it goes into like a train, and then it empties out into the bay. So the function is higher in the oxidation ponds and then decreases and becomes, you know, clean water almost at the end. But we're waiting on the sequences to come in. So this could happen anytime. We're waiting probably next week to take the sequences and be able to put in the composition and diversity aspect of that. I think you're muted, Casey.
Thank you. Had to do that at least once tonight. Thank you again so much, Catalina. We appreciate it. It was a fantastic presentation, and congratulations again.
I'm just so happy to be here. Thank you for having me.
Thank you. Okay, next up, we have chemistry professor, Dr. Claire Till, who arrived at HSU in 2016. From oceanography and analytical chemistry, Dr. Till's primary research is on trace metal concentrations, including copper, iron, and zinc, in the marine environment. Her research has broader impacts on the oceanic environment, which may provide direction for new public policies to preserve the environment for future generations. So far, she has published 19 peer-reviewed journal articles, secured National Science Foundation funding, and the prestigious Cottrell Scholars Award to continue her research at HSU. Please join me in congratulating and welcoming Dr. Claire Till. Hi, Claire.
Hello, thanks so much for that great introduction, Casey.
All right, take it away.
All right. Thank you all for listening today. So my name is Claire Till. I'm in the chemistry department, and I'm going to talk to you about iron and scandium in the ocean. So I want to tell you a story that starts way back in the ancient ocean, billions of years ago, back when there was very little oxygen in the atmosphere. And then there were a couple great oxygenation events where plants were producing enough oxygen so that oxygen started building up in the atmosphere. And that had a lot of changes. Some of the changes were great die-offs from organisms to which oxygen was toxic. Another change is that it allowed the evolution of us and other organisms that need oxygen in the atmosphere. And another change were some massive changes to the chemistry of the ocean. And so I want to start by talking about that. And in particular, iron, which decreases hugely from the ancient ocean to the modern ocean. And this is because iron is insoluble in oxygenated seawater, but it's very soluble in anoxic water. And this is a big change. This is a log scale over here. So this is actually a 10,000 times decrease in the iron concentration from back before there was oxygen in the ocean and to now. So iron is very important biologically. It's in a lot of biological processes because it's good at carrying electrons around. And there's a bunch of metals that are good at carrying electrons around, but iron is kind of the main one that's used most broadly in biological systems. And one of the hypotheses for why iron is so broadly used is because back in early evolutionary history, these microorganisms back in this anoxic ocean were evolving in a time when there was relatively lots of iron. And so they evolved using iron to carry the electrons around. Regardless of whether that hypothesis is true or not, currently we have phytoplankton in an oxic ocean that have a relatively high need for iron, and the ocean has very low iron concentrations. So that leads to some interesting chemistry. So how do oceanic phytoplankton get their iron? They're out there, they're growing. Iron is scarce, but they're still there. So they're obviously getting their iron somehow. So here's some phytoplankton out there. Iron is scarce and essential, but phytoplankton have evolved some really nifty ways of getting it. And one of the ways is secreting organic molecules that bind with iron. And that increases the solubility of iron. So here's an example organic molecule. You see iron in the middle here, a bunch of organic stuff around it. And this prevents, binding with this organic molecule prevents iron from forming iron hydroxides, which is how it would normally precipitate. So it increases the solubility. It's still quite insoluble, but it's more soluble, and the phytoplankton are able to get the iron a little bit better by binding it with these molecules. And overall, 99.9% of oceanic iron is bound to organic molecules and helps keep it soluble. On the analytical side, this makes it complicated for us. Having these organic molecules binding to the iron along with all the other things iron is doing makes it harder for the scientists who are trying to figure out where iron is and how it's shifting around in the ocean and how the phytoplankton are getting it. So let's talk a little bit about understanding the iron cycle in the ocean. So we've known for a long time as a community that iron is really important in the ocean. It controls a lot of phytoplankton growth, an essential nutrient, but often really low concentration. So we've been studying it for decades, but it's still not fully understood because it's quite complicated. So it's got the low solubility in oxic seawater that we talked about. It also binds with a lot of organic molecules, some of which are secreted by phytoplankton to make it more soluble. Another thing that iron does is it binds to sinking particles, and this is called scavenging. So any particles in the ocean, like dust particles or dead phytoplankton that are sinking, iron tends to just adhere to the outside of those particles and then sink away too. So that also decreases the iron concentration available to phytoplankton. There's also a high particulate concentration of iron. So this is like the dust particle goes into the ocean and there's iron in it, but it's not really accessible to phytoplankton, but still it contributes to the cycling of iron and the complexity of figuring out where the iron is and how the phytoplankton can get it. Another thing that iron does is have redox chemistry, which is how it's able to move electrons around in those biological processes, and is part of it being able to be an essential micronutrient. So all these different attributes of iron chemistry weave together and just make it kind of a complex picture where it's harder for us to figure out what's going on. So we could really use some more tools, and that's where we're going. It'd be useful to have a new tool to illuminate iron cycling, and to get there, I want to tell you a specific story about iron being complicated. So here we have the North Pacific Ocean and the North Atlantic Ocean, two specific sites where we have some data for some metals. And so we're introducing two new metals here, aluminum and zinc. And what I want you to notice is that aluminum, so this is purple corresponds with Atlantic and red corresponds with Pacific. So aluminum has higher concentrations in the Atlantic, but zinc has higher concentrations in the Pacific. And this opposite feature has to do with the circulation of the ocean. So this is the great conveyor belt of the ocean. And so you've got deep water forms in the North Atlantic. The blue arrow means deep. And then it flows along and it's still deep when it gets to the Pacific. So the deep water in the Pacific is actually a lot older. It's been deep water for a long time. The deep water in the Atlantic has only been deep water for a short time. And that has implications for the chemistry. So here, thinking about aluminum again, it's a scavenge type metal. And remember, that's where you adhere to the outside of a particle and sink away. So the aluminum is constantly adhering to particles and being removed from the water column. And so over time, as it goes from the Atlantic all the way over to the Pacific, we're losing aluminum as it binds onto particles and sinks away. So by the time we get to the Pacific, there's not very much aluminum left. On the other hand, zinc is a nutrient type metal. So in the surface, zinc is taken up by phytoplankton and used in biological systems. But then if the phytoplankton die and are sinking down, they're slowly redissolving and remineralizing, and the zinc is going back into the water column. So this whole time going from the Atlantic to the Pacific, the zinc is remineralizing and we're getting an increase in concentration. Now iron, because it's got its hands in so many different chemical reactivity pots, does both. And so you see that here we have iron. It's the same in the North Pacific and the North Atlantic. The concentration of iron is the same. And so it's both being scavenged and being remineralized, and both those features are kind of canceling each other out. So this is an example of iron doing lots of different things chemically in the ocean. It's also an example of where our potential new tool comes in, which is scandium, because scandium is also this hybrid type metal, where it's being influenced by two processes, where it's being removed by scavenging, but it also must be increasing in some way too, because it balances out and we end up with hybrid type for both iron and scandium. So this is really exciting to me because the idea is if iron and scandium have such similarities where they're both hybrid type metals, but they're different elements, so they can't be entirely the same, they must have some differences. Maybe we can use the similarities and differences between iron and scandium to start teasing apart those different parts of the iron cycle and start identifying, oh, iron doing this is because of this part of iron's chemistry, and iron doing this is because of this parts of iron chemistry. So let's go back to this diagram, thinking about the different parts of iron chemistry. And now I've color coded it. And yellow represents what iron only does, not scandium. So scandium is much more soluble in oxic seawater than iron. It has a much lower particulate concentration. Scandium doesn't really have redox chemistry, which means it can't move electrons around, which means I don't think scandium is an essential micronutrient. Now, I'm not sure about this. There could be some hitherto unknown use for scandium as a nutrient. Nobody knows about one existing. I'm kind of doubtful because most of the time in biological systems, metals play a role by carrying electrons around. And since scandium can't do that because it doesn't have redox chemistry, I'm doubtful that it's a nutrient. But I put it as light yellow because I'm not completely sure about that fact. Now, on the other hand, scandium and iron both bind to sinking particles, the scavenging thing. So they've got that similarity going for them. The thing is that that's not enough to make scandium a hybrid type metal. If it was just scavenging, and that's the only thing that iron and scandium had in similarity, then they wouldn't both be hybrid types like we were just looking at. So they must have more similarities than just scavenging. So I'm wondering if scandium binds with organic molecules, perhaps the same exact organic molecules that iron binds with. So does scandium bind with the organic molecules iron does? I think this is likely. Scandium and iron are very similar size. And so a molecule that binds iron, it's totally conceivable that you could substitute out iron and put scandium in and it would fit just perfectly. So hypothesis one, it does bind with the same organic molecules that iron does. And that's why scandium and iron have a similar distribution in the ocean. That's what connects scandium to the biological side of the ocean, even though we don't think it's a micronutrient. And my hypothesis number two is we can use the similarities and differences between scandium and iron to tease apart the iron cycle and start to understand more about what's happening with iron and what makes it available for phytoplankton. So I want to end by giving you some preliminary data showing an example where I think we are seeing some illumination on the iron cycle using scandium. And we're doing lots of research in my lab on this right now, so I'm glad to give you more hot off the press stuff at any point if we run into each other and you'd like to hear about it. So here's some example preliminary data. This is where we sampled right off the coast of California, this little X right here. And this is Cape Mendocino, and this is San Francisco Bay. And so we're just right off the coast. And so this is a depth versus concentration. The bottom of the ocean, right where we were sampling, is just 95 meters or so, so right beneath where we were. Scandium is in orange, iron is in blue. So, observations, both scandium and iron increase in concentration near the bottom. They're going up in concentration near the bottom, but iron is increasing more than scandium, relatively more than scandium. Couple possible causes for why iron and scandium are increasing with depth. One of them is resuspension of sediment containing iron and scandium. So right down at the bottom here, there's a bunch of sediment, and just water moving over the top of the sediment can bring some of that sediment into the water column. A lot of the sediment contains metals, so it could bring up iron and scandium with it. Another possible source is redox chemistry in the anoxic sediment increasing iron solubility. So remember back in the ancient ocean, it had anoxic seawater and iron was supersoluble. In the sediment, there's anoxic seawater and iron supersoluble. So we get tons of iron dissolved right beneath the sediment. And if some of that pore water comes out into the water column, suddenly we have more iron in the water column. Now it's about to precipitate and sink out, but we're probably catching it while it's still resuspended in the water column. So these are two possible causes, but only one includes scandium and iron, and the other is just iron. So I'm wondering whether scandium can tell us how much of the iron signal is from resuspension, and then the additional iron signal that we're getting is coming from the redox chemistry. So scandium may tell us how much iron is from each of these possible sources and may help us figure out which part of the many aspects of iron chemistry is responsible for what we're seeing in one place versus another place. So scandium could be a new tool to help differentiate iron's complex cycling, and that's what we're working on in my research group right now. So thank you very much for listening. I have here some collaborators and students and my funding sources, and I'd be glad to take any questions.
Thank you so much, Claire. A couple questions from the audience. One is, I've heard that you have to keep a very clean lab for your research. What does this entail?
Okay, great question. Yeah, so we have HEPA filtered air. You've heard about HEPA filters probably recently on airplanes and stuff. We have HEPA filtered air for our lab. And so the air is coming in through the HEPA filter, and we have the room be positive pressure so that that's the only way the air is coming in is through the HEPA filter. And then we're also wiping down surfaces all the time our enemy is dust, basically. And then the other thing we do is we use a lot of plastic, because plastic you can clean really well for metals, and glass, no matter how much you clean it, it's not going to be clean for metals.
Nice. And what field work do you have planned as a continuation of this project?
Great question. Yeah, so this summer we're going to be sampling from some rivers. There's some really interesting organic molecules binding iron in rivers, and that's a really well-established pathway. And so now we're going to see if scandium follows the same pathway, because maybe it binds with the same organics. So we're going to sample some local rivers right where it goes from fresh water to salty water, and that's right when the iron precipitates out, and we'll see if the scandium does too.
Wonderful. Thank you so much, Claire. Again, that was a wonderful presentation, and congratulations on being named a McCrone Scholar.
Thank you so much, Casey.
Okay, last but certainly not least, we have Environmental Studies Professor Dr. Deepti Chatti, who brings a unique set of interdisciplinary research skills to HSU. As a critical social scientist of energy and the environment, Dr. Choddy conducts ethnographic research in India and the United States. She serves as an affiliate faculty member in HSU's Environment and Community and Energy and Technology and Policy graduate programs, and is also a faculty research associate in the Schatz Energy Research Center. In her teaching and research, she focuses on global challenges like clean energy access, sustainable development, climate justice, and public health. An active scholar, Dr. Choddy is currently working on a book manuscript and published 5 journal articles and 2 book chapters in recent years. Please join me in congratulating and welcoming Dr. Deepti Choddy. Hi, Deepti.
Hi, everyone. Can you hear me all right?
You're perfect. Take it away.
Okay, awesome. I'm going to quickly share my screen. Okay, can you see that? Okay, I hope you can see that. All right. Thank you so much, Casey, for that generous introduction. And thank you to everybody at HSU and SPF for recognizing my scholarship, especially all my colleagues. Thanks to Dr. and Mrs. McCrone for being here today and listening to all of our scholarship. And then last but not least, I'd like to say a quick hello to my family and my partner who are calling in from different parts of the world, India, labs in UCSD, and so on. So my talk today is called Changing Hearts and Minds in Rural India, Energy Access, Air Pollution, and Environmental Justice. A little bit about me, as Casey mentioned, I'm an interdisciplinary environmental scholar and teacher, and I primarily use qualitative research methods to study energy transitions, clean energy access, and exposures to air pollution in historically marginalized communities. For about 10 years, I've been carrying out research in India, and more recently, I'm starting research here in the United States. A lot of my work is collaborative, so I have active research collaborations with scholars from different disciplines, as well as community-based organizations in India and tribal governments here in California. That's the new project that we're starting here in Humboldt County. So what do I study? So I study things that are typically thought of as being in the realm of environmental justice, alongside questions that are typically thought of as being in the realm of sustainable development, with a critical analysis of science and technology. And the topics that I study are things like energy access, public health, climate change, women's work, agency, and livelihoods. A quick outline for my talk today. I have a lot that I want to share with you, so I'm going to speed through it, but I welcome your questions at the end. First, I'll give you a brief background on household air pollution, sustainable development goals, and energy access. I'll give you a brief overview of recently completed research that I carried out between 2013 and 19 in India. I'll give you a tiny excerpt from one chapter of the book project that's resulting from that work. And then I'll end with some theoretical contributions and larger intellectual questions of my work. So, air pollution is known as a silent killer. Public health experts estimate that every year around 7 million people prematurely die from air pollution. Just to put that number in context, in 1 year of the ghastly and tragic COVID-19 pandemic, we had approximately 2 million people die. So you get a sense of what scale of magnitude air pollution does in terms of its mortality and morbidity. This is a graphic from the World Health Organization. And in the bottom half of the graphic, what you see is where the spatial distribution of where this premature mortality and morbidity is. This is an environmental justice issue, as you can see that most of this death and ill health happens in low and middle income countries. And that the part of the world that's shown in black, which is South Asia, is where I focus most of my work. Air pollution is actually the #1 risk factor in India, ranked by burden of disease. This is data from the global burden of disease. And so it's really an important issue to study from an environmental health and justice perspective. Now, typically when we talk about air pollution, all of us tend to think about outdoor or ambient air. You know, we've all read headlines about New Delhi's air or Beijing's air or Los Angeles's air. But what I focus on is household air pollution or indoor air pollution, which accounts for more than half of the deaths that I mentioned to you earlier. And this is predominantly caused by the incomplete combustion of household cooking fuels, solid biofuels in the form of wood, crop residue, animal dung, and so on. Now, how is this related to the Sustainable Development Goals? Well, it focuses directly on Sustainable Development Goal 7, which says that by 2030, we aim to ensure universal access to affordable, reliable, and clean and modern energy services to everybody, which has two components. On the one hand, this is about expanding infrastructure and changing and upgrading technologies. And on the second aspect of it, it's about substantially increasing the share of renewable energy within the global energy mix. So I'm going to jump into the overview of recently completed work in India. This work basically critically analyzed the epistemic, ethical, and empirical challenges of sustainable development in the global south. And some of the questions that the people that I was studying were asking, how do we expand energy access and facilitate a clean energy transition and reduce exposures to air pollution in the context of global climate change? The photograph that you see here is of what's called a so-called traditional stove or a mud stove. And this is the primary cooking technology used by 40% of the world's population. So most of the world's poor use cooking technologies that they make themselves out of locally available materials and combust fuels that they access locally in their region, such as that you see here. And as you see the air pollution on the wall, that's indicative of the kinds of smoke that we're talking about. So in order to study this question, I conducted 18 months of multi-sited, multi-scalar ethnographic research across various parts of India, indicated by the blue dots over here. And these sites straddled rural homes, cook stove companies, international aid organizations, government offices, and academic and policy research organizations as well. And because I'm interested not only in critically analyzing the development projects that attempt to intervene in lives of the poor, because I'm also interested very much in studying the way that knowledge is produced about air pollution, climate change, and poverty alleviation, I ethnographically studied one particular transnational cook stove research project that was funded by a federal agency and an international aid organization and unfolded for 4 years from 2013 to 17. Now, this project was structured as a randomized control trial in 2 states in India, indicated here, Himachal Pradesh and Karnataka. And I was the resident social scientist of this project, so I got an inside view into how these things work. So just to summarize, I have multiple ethnographic interlocutors. On the one hand, I'm working with individuals and families whose kitchens are the sites of intervention and whose everyday energy decisions are coming under scrutiny for a variety of public health, environmental, and social reasons. The second set of interlocutors are development actors, state and non-state, who are trying to facilitate an energy transition, and then academic and policy researchers who are trying to create knowledge out of all of this that's going on. And just a quick look at, some of these faces might be familiar to you. These development actors are of all kinds of scales, of all kinds of networks, including private companies, international aid, as well as national governments. And a quick look at the technologies that are supposed to be the so-called advanced cooking technologies that are supposed to replace the mud stove that you saw earlier. This is just a snapshot of a whole umbrella of technologies that come under this category. Some of these still use solid fuels, and some of them use other kinds of fuels like propane, which in India is called liquefied petroleum gas, or LPG, as well as electricity. So I'm now going to give you an excerpt of one chapter of the book, so you get a flavor for it, and hopefully that makes you want to buy it in a few years when it comes out. So this chapter is called Placing Kitchens in a Good Life. And in this chapter, in this excerpt, what I reflect on is the idea of change in rural Himachal Pradesh. And I do that by focusing on home improvements that are routinely carried out by rural families, as shown in the photograph here. Now, this version of rural life is obscured in energy transitions literature. Within this literature, rural families are imagined as being resistant to change. And so I contrast this version of rural life, which is constantly dynamic and in flux, with the idea of an energy ladder, which is a teleological understanding of energy transitions. So the energy ladder, some of you might be familiar with this. It operates on the assumption that an increasing wealth in a family leads to a climbing up of the ladder and facilitates an adoption of new technologies and a disadoption of old technologies. But most families actually do not stop using old technologies when they adopt new ones. This is widely recognized, and this phenomenon is called stove stacking. So in rural Himachal Pradesh, where this excerpt is based from, multiple cooking technologies in kitchens are actually the norm. And most people want multiple stoves. Now, this is for several reasons. On the one hand, you have pragmatic considerations. For example, you have cold winters, which require wood stoves, and those kinds of stoves called tandoors, and I'll show you a photo in a second, serve the dual purposes of space heating as well as cooking in the winter. You have problems of access of clean cooking technologies and fuels. For example, in the monsoon or in winter, a lot of the villages are cut off from more urban parts of the state. And so any fuel that requires you to go into market to refill is simply not feasible in those kinds of weather conditions. And of course, there's a pragmatic consideration of expense, that it requires a market-based exchange. But these are not all. There are also aspirational considerations to consider. And that's what I'll focus on now. So in order to dive into what the aspirational considerations are, I am going to present to you some qualitative data that I collected from my rural interlocutors by essentially, I asked them to draw what their dream homes look like. So what I'm showing to you here is Monica's dream home. Now, Monica is a pseudonym per ethnographic convention, I'm anonymizing her. And there are several things to notice about Monica's dream home. To make it easier for you, I've drawn some arrows and some photographs so you can follow along. Now, even in Monica's dream home, please notice that there are multiple kitchens and multiple stoves. And now this is directly contrary to the external imaginations of modern homes, where it's assumed that there'll be one kind of kitchen and one kind of stove, which is the LPG propane stove. And in Monica's dream home, she has her, the K1 kitchen has the mud stove that you see on the upper right corner of the page. K2 has the tandoor, or the wood stove that I mentioned, that serves the dual purposes of space heating and cooking. And K3 on the bottom right is her LPG gas stove. So Monica wants all of these in her dream home. Now, I want to show you a diagram that Shruti drew of her dream home. And, similar to Monica's dream home, there are multiple kitchens, multiple stoves. But what's also really striking about Shruti's dream home is how different her drawing is from her actual home. In her actual current home, she only uses the LPG gas stove for cooking. So to an external observer, Shruti has adopted LPG and left her mud stove behind in the past. So one could imagine her well on her way to the upper rungs of the energy ladder. However, the irony is that Shruti considers herself to be in a temporary period of hardship when she's unable to have the luxury of a mud stove. So as she pours her labor and resources into cash crops, hoping to cash in on a bumper harvest, her plans include expanding her home to build a proper kitchen with a mud stove. She would often say to me that after a few years, हम अच्छे से बनाएंगे, gas भी होगा, मिट्टी का चूल्हा भी होगा, tandoor भी होगा, which roughly translates from Hindi into, we will build it well, there will be gas, there will be a mud stove, there will be a tandoor. So what does this all mean? I'm suggesting that in order for us to engage in any forms of ethical practices when we are engaging in sustainable development projects or questions of environmental justice, that we need to actually center emic conceptions of what people think is a good life to aspire to, and that any energy transitions work has to actually be informed by emic perceptions of good health and well-being. When I say emic, I mean which are the perceptions of the targets of development. So in other words, what my book is going to be contributing to is theorizing what energy transitions look like from the vantage point of the kitchens, of low-income kitchens in rural India. And so if there's one takeaway that you take from my work today, it should be that even well-intentioned sustainable development projects can propagate environmental social injustice by not allowing for the plurality of epistemic frameworks and not recognizing the multitude of priorities that are already entangled in the sites of development. A quick thank you to all the organizations that have resourced much of this work, and I really welcome all your questions now.
Thank you so much, Deepti. That was fantastic. One question from the audience: are solar stoves a viable option?
That's a great question. And, a lot of people over decades have tried to introduce solar stoves. And I think one of the things that I've realized is that there's really not one technological silver bullet for these complex set of environmental, social, and health issues. So I think there are certainly places where solar stoves can fill a niche need, but I think that I doubt that they would take over all of the cooking needs of a household.
Wonderful, thank you. And another question: why did people always draw the mud stoves in the attic?
Oh, that's a great question. So another part of my book actually looks into the spatial organization of people's homes and looks at why people place certain things within the home where they do. And this has to do with several things. On the one hand, the mud stove is the smokiest stove. And so having it at the top of the house where there's lots of opportunity for the smoke to escape from the porous roof allows the smoke to just leave the house without going into the other spaces of the home. There's also a spiritual dimension to it. In rural Himachal Pradesh, a lot of the families believe that their family deity resides within their mud stove, and elevating it at the top of the house is actually sort of a common practice. It's seen as being associated with that. So there's all kinds of reasons for that, really.
That's beautiful. Thank you so much, Deepti. And again, I want to congratulate you on your award tonight and for presenting. That was fantastic. So appreciate it.
Thank you so much.
All right. So as always, thank you again for everyone joining and zooming in with us. And we love any opportunity to share the amazing research and creative works happening at Humboldt State University. And we just couldn't be more proud of our award recipients this year. If tonight's program got you inspired and wanting more, I hope that you will all join us next week for Idea Fest, which is Humboldt State University's annual research symposium, which kicks off on Monday, May 3rd. We have an entire week devoted to sharing more scholarly and creative works and research projects of our students, staff, and faculty. So tonight was just the beginning. Congratulations again to our 2021 McCrone faculty and student scholars. I hope everyone has a wonderful rest of your week and a very good night. Thank you all.